Hello and welcome to all my dear students. So far, we have studied in this chapter about one gene interaction, two gene interaction, polygenic, pleiotropy, so many things. I hope you all have understood that well. Now, today we are going to start with the next topic in the chapter, that is linkage. Beta, if you remember one thing, I told you that Mendel, while preparing his dihybrid cross, he proposed a law called law of independent assortment, according to which when two genes they are inherited together from located on homologous chromosome, then both the genes during the time of gamete formation they segregate out independently, followed by their alleles. But actually it is not so. Two genes located on homologous chromosome, they do not tend to segregate out independently. Rather, one gene influences the other gene, right? And this thing was not understood by Mendel. Mendel could not explain it. However, when T.H. Morgan, scientist, the father of experimental genetics, while experimenting on Drosophila melanogaster, he found that two genes when inherited together, they do not tend to separate out independently, rather they influence each other, and he coined the term linkage, which is an exception to Mendel's law of independent assortment. Clear? So what I am saying, let me repeat it once again so that things become more clear. Beta, while studying dihybrid cross, Mendel proposed law of independent assortment, to which an exception is linkage, as according to Mendel, when two genes located on the homologous chromosome, they tend to segregate out independently without influencing each other during the time of gamete formation. However, it is not so. Two genes, they do influence each other if they are present on the chromosome, if they are present on the same homologous chromosome. And this was explained by Morgan under the heading linkage. Correct? So now let's try to understand the experiment performed by this fellow Morgan, and let's try to understand what actually linkage is, whether it is useful or not. So now we are going to start. Today's class is all about understanding the concept of linkage and recombination. So first point itself is written on the screen that T.H. Morgan worked on Drosophila while working. Now, he worked on Drosophila, right? You know that Drosophila is one of the best organism to study for the study purpose. All the points I have already explained in my last class. Now, one of the reasons of choosing Drosophila as an experimental organism is that the number of chromosome in Drosophila cell is very less, which is equivalent to 4 pair, out of which 3 pair are autosomes and 1 pair is sex chromosome. Drosophila beta, they show same sex determination pattern as that of humans, that is XX means female, XY means male, right? So this fellow Morgan was studying actually on the sex chromosome of Drosophila. And which sex chromosome, whether X or Y? So this fellow Morgan was studying on the genes located on X chromosome, located on X chromosome. Correct? Now, which 3 genes he took? He took those 3 genes controlling the body color of Drosophila, eye color of Drosophila, and wing size of Drosophila. So these are the 3 characters chosen by Morgan, and all these characters, their genes are located on X chromosome. So basically, Morgan was studying on the sex chromosome to discover the concept of linkage, right? Now, you know, Mendel suggested one thing, that whenever you have to begin the cross, the parents has to be always pure line. One parent has to be dominant pure line, another parent has to be recessive pure line. So whenever any scientist try to start this hybridization program, they always follow the words of Mendel. Always the parent chosen are of pure line nature. Correct? So now let's see that each character is having two traits, you know. So basically Morgan studied 3 characters having 6 traits. Traits means the quality of the character in dominant state and in recessive state. Correct? So body color is controlled by gene Y. Body color is controlled by gene Y located on X chromosome. The dominant is this. Plus means beta dominant is also known as wild type. Dominant is also known as wild type trait, whereas recessive is also known as mutant type trait. So the dominant body color, homozygous dominant body color is brown, recessive is yellow. Correct? Coming to eye color, the dominant, the wild type color is red, which is controlled by gene W. When you beta mention plus over the gene, that means you're talking about dominant, and without plus means recessive or mutant. Plus means dominant, no plus means recessive. Correct? Now wing size, big or normal. And miniature. So miniature, it is controlled by gene M. So M plus means homozygous dominant, whereas small m without plus sign means homozygous recessive. So these are the 6 traits which are located on X chromosome, controlled by genes like Y, W and M. And these are the 3 characters which were chosen by Morgan to perform his cross. Now with the help of these 3 characters, this Morgan prepared 3 crosses. Morgan prepared beta how many crosses? He prepared 3 dihybrid crosses. How many crosses he prepared? With the help of 3 characters, he prepared 3 dihybrid crosses. One cross was made between these 2 characters, that is body color and eye color. Second cross was made between these 2, that is eye color and wing size. And third cross was made between body color and wing size. So these were the 3 dihybrid crosses made by Morgan in order to understand that whether if 2 genes are inherited together from the homologous chromosome, so do they separate out independently or they do influence each other. Okay? Understood so far? Chalo. Now let's try to see the cross made by this fellow. So first of all, to start the cross, you have to select the parents, and parents are always pure line. Now, our parents are pure line. So one we are going to take male and one we are going to take female. Correct? Now, male is determined by XY type of chromosome, whereas female is determined by XX type of chromosome. So in female, both the chromosome are same. So their representation is also going to be the same. So this is my female chromosome, female parent, which is crossed with male parent. Now you know, in male, one is X chromosome, another is Y chromosome. So obviously they are different chromosomes, so their representation has to be also different. Now you see how I represented Y. Y, we have just made a slight slant. There's a straight line and then there is a slant that denotes here that it is a Y chromosome. Correct? Now this becomes my male parent, female parent and male parent, pure line parents are ready. Okay, now tell me beta, the genes are located only on X chromosome, right? That means female will be either homozygous or heterozygous for the gene. However, males are only going to be hemizygous for that gene, as males are having two different sex chromosome. One is X, another is Y, right? Chalo, see. So over here, we have chosen homozygous, pure line parents, dominant as well as recessive. Suppose my female is homozygous dominant for the two characters, that is body color and eye color. So how will I represent? Yes or no? So these are my parent. So this female is homozygous dominant for both body color and eye color. Can you see? I have represented with Y plus, W plus, W plus. Right? Now here my male is going to be homozygous recessive for the character, but because you know it is having one Y chromosome, hence the condition which you have to show over here is hemizygous. So of the gene, only one allele will be present. Understood? Now, these are the parents. Now it's the time to prepare the gametes for first generation offspring. Now beta, gametes of F1 generation. Can you see? It's what? Homozygous dominant for both the gene. So number of heterozygosity is 0. 2 की power 0 means only one type of gamete will be produced, and that gamete will be having both the dominant alleles of the gene. So this is the gamete which is contributed by the mother. Now, in case of father, how many gametes will be obtained? 2. One having X chromosome, another having Y chromosome. So these are the two gametes obtained from the father. Now what will happen? What will happen? Fertilization will take place. 50-50 probability that this female gamete may fertilize with this male gamete or with this male gamete. So there's probability is 50-50%. Hence, F1 generation offsprings. F1 generation offsprings are expected to be females will become heterozygous dominant, correct? Whereas males will become hemizygous dominant. Yes or no? So here all females of F1 generation beta will have brown body with red eye color. Yes or no? But heterozygous. So this is the genotype and this is the phenotype for F1 generation females. Whereas all males of F1 generation will also become same. Will have brown body and red eye, because now they have become dominant. They're having the dominant alleles of the gene. Initially they were having recessive alleles. Now due to the combination, now they are having the dominant alleles of the gene. So they will have dominant feature. Okay. So everything till here is normal. Mendel also said the same, that whenever pure line parents are crossed, when pure line dominant male or female is crossed with pure line dominant or recessive male or female, then all offsprings, they are bringing the dominant phenotype. So here also same thing is happening. You can see mother is dominant, father is recessive, but all the offspring, be it male, be it female, all are having dominant phenotype. No recessive phenotype, no offspring with yellow body or white eye or white-eyed coloration. So till here everything was perfect according to Mendel. But now Morgan continued the cross and he tried to perform selfing. Correct? So now next stage is to perform selfing. But before selfing, we have to check the gametes for F2 generation. Chalo. Now just see beta. Gametes of F2 generation. Clear? Chalo, let's go back. So this is my mother, right? Now, which she is heterozygous for both the gene. Can you see? Y is also heterozygous and W is also heterozygous. That means, what will you say? You will say that number of gametes produced from this female will be 2 ki power 2, which is 4 gametes. And what will be the 4 gametes? 2 parental, 2 recombinant gametes. Correct? So recombination will also take place. Exchange of genetic material. Right? So total number of from female, how many gametes we are going to obtain? 4 gametes. And what are they going to be? 2 parental. Check this. Just check this. 2 parental, right? And 2 are the recombinants. So these 2 are the parental gametes and these 2 are the recombinant gametes. Clear? Now from the father, now this is also going to produce the gametes. However, you know here crossing over cannot take place because X and Y, they are partially non-homologous. Now this Y allele is not having its partner allele. This W plus allele is not having its another partner allele. So recombination cannot take place. Hence again only 2 types of gametes will be produced. One dominant allele, one which is having X chromosome, and another gamete which is having Y chromosome. So these are the gametes contributed for F2 generation by the male parent, and this is contributed by the female parent. Now what will happen? Gametes are ready. Let's try to produce the zygote. How many zygotes are possible? 4 gametes from female, 2 gametes from male. So 4 twos are 8 zygotic combinations are possible. 8 type of offsprings can be obtained from this cross. Chalo, now let's see. So now F2 generation, gametes are obtained. For this we have to prepare a Punnett square. You know the rule. Everything see is going according to Mendel only. 8 combinations we want. Okay. Now, here I am going to write my female gametes, and on this horizontal line I am going to write my male gamete. So, male only contributed 2 types of gametes. One with X chromosome, one with normal Y chromosome having no alleles of the gene. Right? But female contributed 2 parental combinations of gamete and 2 recombinant type of gamete. So these 2 are recombinant, these 2 are parental. Any confusion to anybody that how to decide the parental gamete and how to decide the recombinant gamete? This I have already taught you while I was teaching you the dihybrid cross of Mendel. Right? Chalo, now just fulfill the cross. Okay. So how many type of zygotic combinations are possible? 8 type. Just let me just fulfill this block and then I will explain. Check this. Everyone on the screen. Now these 4 types are female and these are 4 types of male. right? Now you see beta, the first four are parental zygotic combinations. Yes or no? The first four are parental zygotic combinations for F2 generation, whereas the last four are the recombinant zygotic combinations for F2 generation. Yes or no? Yes. Now, okay. This everything is according to Mendel. Now, Mendel said the ratio of parental is to recombinant for dihybrid cross at F2 generation has to be 9:3:1. This has to be the phenotypic ratio. But unfortunately, the ratio got highly varied. The chances of parental zygotic combination was 98.7%. That means whatsoever offspring was coming out in F2 generation was having parental combination only. 98.7% of the offsprings in F2 generation were of parental type, whereas only 1.3% of the offspring were having the recombinant types of combination at F2 generation. Where 98.7, where 1.3, there is no match. It seems now this Morgan thought that if only parental combination offsprings are coming, that means crossing over is not taking place. The chances of formation of these gametes is very, very low. Only parental type of gametes are being produced in F2 generation. Understanding or not? See, what Morgan said, this is how the genes located. Right? The two heterozygous genes are located like this. Now, the parental combination means Y plus W plus or YW. Now, this is one gamete, this is another gamete, two types of gamete. Now, according to the observation of Morgan, he saw that 98.7% of the offsprings, they are bringing only the combinations exactly of parent. They are having this type of gametes. That means recombination, crossing over is not taking place. The percentage of formation of these gamete is less than or is near about 1.3% only. This means that crossing over is not taking place or is taking place very minimally. Very less crossing over is taking place. That is why chances of parental zygotic combination at F2 generation is very high, and formation of recombinant zygotic combinations at F2 generation is very, very low. This means that these type of gametes are not forming or forming at a very, very low rate. Understanding? Yes. Okay. Now beta, hence this fellow Morgan got confused that why only parental gametes are being produced, why not recombinant gametes, where are they? So he found that these two genes, they are located on the same chromosome, which is X, and they are very, very closely present. As a result, they are not able to perform crossing over. Such genes which are located very close to each other, they are called as linked genes. Now these two are beta called as linked genes. Linked means they got associated, they got held together, because of which they are not allowing the exchange of genetic material. This Y plus and W plus and Y, they are not able to meet. because y plus yw, they are holding each other very tightly as the distance between the two genes is very, very less. Such genes are called as linked gene. Now you will think that, ma'am, Mendel also chose 7 characters. His characters controlled by genes were also located on same chromosome. Then why Mendel did not got this linkage? Because beta, Mendel studied those genes controlling those character located on same chromosome but very distantly. Dur dur the, they were not closely present. Now suppose over here, one gene is present over here, another is present over here. They are on the same chromosome, but here there will be no linkage between them. They will not be called as linked gene, right? They will produce the gametes same as that of Mendel, 9331 ratio, that is 10:6 ratio, correct? Why? Because the distance between the two genes is high. Understanding my point, everyone? So Morgan suggested one thing like this. He said that if two genes are located on So he said that if two genes are located on same chromosome and if their distance is less, then chances of crossing over decreases. Then chances of crossing over decreases, thus the rate of linkage increases. Clear? Thus the rate of linkage increases. Understood or not? Okay, very good. Now, let me just add one more page, or it's okay. On this basis only, student of Morgan, that is Alfred Stewart Went, tried to locate genes on the chromosome. He tried to map the genes on the chromosome because it is said that genes are located linearly on the chromosome. Since genes are located linearly on the since genes are located linearly on the chromosome, my dear, Alfred Stewart Went tried to map means locate the genes on the chromosome right on the basis of distance between them. Distance between them. Correct? It is said that distance between the two genes is inversely proportional to the percentage of recombination. More the distance, is directly proportional to recombination. That means more the distance, more will be recombination, lesser will be linkage. So directly proportional to recombination. Recombination beta means crossing over, exchange of genetic material. Or you can say inversely proportional to linkage. More the distance, lesser the linkage, more recombinant gametes, more recombinant zygotes. So this is the relationship suggested by Morgan, right? And now how Mendel could not get this linkage? Because obviously the genes which he chose, the character which he chose, even if they were located on the same chromosome, their distance was much more than कि उनको linkage नहीं मिली. He could only get proper parental type and proper recombinant types of gametes. Correct? Yes or no? Now will you all remember this relationship? So this is very, very important, and this is the gist of the entire experiment performed by Morgan. Now one more thing, percentage I told you. Now in you know that Morgan beta performed 3 dihybrid cross. One dihybrid cross I have already drawn on the screen, which you can see very clearly over here, right? And the percentage of parental and recombinant type for his first dihybrid cross was 98.7%, and recombinant was very, very less, which is only 1.3. The second dihybrid cross which he performed was between the eye color and wing size, where he found that the percentage of parental was 62.8% and recombinant was 38 point, now you can just do the remaining calculation, 62.8%. Now if I want to add this 37.8, sorry, 62.8 and 37.2%. So this is the combination of parent and recombinant zygotic combinations at F2 generation of his second dihybrid cross. Correct? Now the ratio of the third dihybrid cross you will tell me when I will tell you the chromosomal mapping concept. Chalo. Now according to Alfred Stewart Wendt, who is the student of he was the student of Morgan. So Morgan was basically working on linkage. Now this Alfred Stewart Wendt said that if genes are located linearly means in a row, in a line, one after the other, and if their locations are fixed, correct, then why can't we say that the percentage of recombination between the two genes is equivalent to the distance between the two genes? That means if this your body color gene is Y, right, and eye color gene is W, the percentage of recombination between them was 1.3%. So according to Stewart Wendt, this percentage of recombination between the two genes is actually the distance between them. And the unit for prokaryote, the distance of the two genes for the unit is centimorgan. So according to him, Y and W, they are located at 1.3 cm, which is very, very close to each other. This is how chromosomal mapping can be done by calculating the percentage of recombination between the two genes located on the same homologous chromosome. Clear? Yes or no? So according to beta Alfred Stewart Wendt, since genes are located linearly on the chromosome, distance between the two gene is equivalent to the percentage of recombination. Correct? That means, for example, Y gene and W gene on X chromosome of Drosophila, they are located 1.3 centimorgan far away. Now, second, W gene, that is eye color, and wing size gene on the same X chromosome, that is M gene, they are located 37.8 centimorgan. Check it. Where is it? Yes, see, 37.2 centimorgan. 1.3 is the distance between these two genes, and between these two genes, the distance is 37.2. Now, if genes are located linearly, then beta, what will be the distance between Y and M? You just have to add on 37.2 plus 1.3. So now that means all the three genes are located linearly on the same line. Correct? That means how can you say? Check it this way. Just a second. Let me add a page. Okay, now just see. This is suppose my chromosome. Three genes are located on the chromosome. Right? Three genes are Y gene, W gene, M gene. Y gene controlling the body color of Drosophila, W gene controlling the eye color of Drosophila, M gene controlling the wing size. Now, distance between these two is 1.3. Distance between these two is 37.2. Now, automatically distance between Y and M will become, because you know genes are located linearly one after the other. So, it will be 37.2 plus 1.3, which is 38.5 centimorgan. Now, automatically you can tell the percentage of parental gametes and percentage of recombinant gametes for the third type, for the third dihybrid cross performed by Morgan. Yes or no? That means for Morgan's third dihybrid cross, for Morgan's, which is the third dihybrid cross, beta? The cross which is performed between body color and wing size. The percentage of recombinant gamete is only 38.5. Now, parental you can do 100 minus 38.5. You will get the parental as well. Easy? Understood what is linkage and how it is an exception to law of independent assortment? Chalo, very good. So, this much is there for linkage. Now, we are going to move one step ahead in the chapter, that is sex determination by Henking. Now, I used this term sex determination previously also, while I was telling you about the sex determination pattern of Drosophila and human. In this, XX is for female, XY is for male. Now, who told you? Who told you that X chromosome is this, Y chromosome is that? Someone must have discovered this concept of sex determination, this XX, XY type and all that things. So yes, that we are going to study now. I am not going to teach you complete today. Yes, but I am going to just give you a hint about sex determination. Later, major portion of this concept of this topic we are going to discuss in our next lecture. But before I say bye-bye to all, let's study little bit information about sex determination. Sex determination concept was given by scientist Henking. Right? Now this Henking beta, he was actually studying the spermatogenesis. What is spermatogenesis? Sperm formation in some insects. So this fellow Henking was studying the spermatogenesis in some insect, where he observed that two types of sperms are produced. Right? Two types of sperms are produced, out of which 50% are receiving a special body called X body. Actually, while he was studying spermatogenesis, he was actually focusing on only one specific chromosome. Right? He was just focusing on specific one structure, and he saw that that specific one structure is received by only 50% of the sperm, and 50% of the sperm are not receiving that specific structure which is studied by him. So 50% are receiving, and that structure by him is called as X body, whereas 50% of the sperm did not receive it. Later on, this X body was called as X chromosome. So X chromosome was basically decoded by Henking, and later on Steven scientist said that the remaining 50% of the sperm which are not receiving X chromosome are actually receiving the second type of male chromosome called Y chromosome. So Stevens discovered Y chromosome. Like this, people came to know that in some insect, males are having combination of sex chromosome as XY. Then later maybe oogenesis was also studied. Then it was found that in females, all the gametes are receiving same type of sex chromosome, that is X chromosome. That means the combination is XX. Like this concept of sex determination came into existence. Then for different organisms, the sex determination was done, and we came to know that different types of organisms are having different types of sex chromosome. However, it was initiated primarily by Henking while he was studying spermatogenesis, formation of sperm in some insect, where he found that 50% of the sperm are receiving X body and 50% are not. The X body itself is the X chromosome, and the 50% of the sperm which are not receiving X chromosome are actually receiving the second sex chromosome, which is Y. Correct? Yes or no? This Y chromosome was discovered by Stevens. So this much you have to understand in the basic concept of sex determination. Bachcho, this sex determination is done on the basis of chromosome. So you can say chromosomal sex determination. However, in some organisms like turtles, in like your crocodiles, sex determination is not done by chromosome. Rather, it is environmental. Temperature plays the important role in deciding that whether the egg on hatching will be male or female. So sex determination can be done by two ways. One which is very, very common, that is chromosomal. Like if I am having XX chromosome in my zygote, so I'll be a female. If in my zygote combination is XY, so I will be male. That means in humans and in many of the organisms, the type of chromosome, the type of chromosomal arrangement is deciding that whether the organism is going to be male or female. But in some, chromosomes are not deciding. It is the environment, it is the temperature which is deciding that whether the organism will be male or female. Correct? Like for example, in turtles, in crocodiles, their sex determination is temperature based, is environmental. Right? Yes or no? So sex determination can be made genetically or environmentally. Environmental sex determination means when sex of an organism is dependent upon the external conditions like temperature. Right? For example, we see this in case of turtles and crocodiles. However, majority of the organism shows chromosomal sex determination. Now, what is chromosomal sex determination? All this detail we are going to start from our next class. Today you just have to understand and revise and solve the questions from your linkage only. I hope you all are going to do that. Yes or no? So, I'm going to just finish my today's class over here. All these things we will do later. Now, beta, meanwhile, you all can do the homework which is given to you, right? And yes, sex determination things, question you can leave. Once I will complete the topic of sex determination in my next class, then surely you all can solve the questions as well. So till then, thank you for joining me, for seeing the class, enjoying the class, understanding it. See you in the next session. Bye-bye everyone.